All right, let's dive right into one of these central mysteries in neuroscience, Parkinson's disease. In this explainer, we're going to play detective. We're going to follow the evidence from the symptoms a person feels all the way down to the molecules to really unpack the role of the prime suspect in this case, I think called the Lewy body.
So to kick things off, let's start with a number that's pretty staggering, 80%. As in, up to 80% of the brain's dopamine-producing neurons, these incredibly important cells, can be lost before someone even starts to show the classic motor symptoms of Parkinson's. Just think about that. It's a massive, silent takeover happening long before the alarm bells go off.
And that right there is the fundamental question driving so much research. What is causing this devastating neurodegeneration? If we can get to the bottom of that, if we can understand the driver, then we can start talking about real therapies that could slow or maybe even one day stop this disease in its tracks.
So here's our game plan. First, we'll piece together the clinical puzzle. Then we'll zoom in on the key pathological hallmark, our prime suspect. We're going to break it down, see what it's made of, track its movements through the nervous system, and finally, look at some of the genetic and cellular accomplices involved in this whole story.
Okay, first things first. Let's establish the scene of the crime. You have to understand the clinical picture of Parkinson's to even know what clues you're looking for in the brain.
Now, we all know Parkinson's for its motor symptoms, right? The slowness, the tremor, the rigidity. But what's really fascinating for us as investigators is this whole host of non-motor issues. Things like losing your sense of smell, constipation, sleep problems. These can show up years, sometimes even decades, before any tremor starts. And that right there is a massive clue that this isn't just a problem in one tiny part of the brain. It's much more widespread.
So if the pathology is so widespread, what kind of evidence is it leaving behind? Well, when pathologists look inside the surviving neurons of a person with Parkinson's, they consistently find one specific telltale sign.
And here it is. This is our prime suspect, the Lewy body. It's basically an abnormal clump of protein that shouldn't be there, found inside the neuron's cytoplasm. Finding these is the definitive post-mortem confirmation of a Parkinson's diagnosis. It's the smoking gun.
And when you actually see them under a microscope, it's kind of wild. They have this really classic appearance, a dense core with a lighter ring or halo around it. It makes them look a lot like a tiny little eyeball just staring back at you from inside the cell. Okay, so what is this thing actually made of? It's really important to get this. Lewy bodies aren't just a random pile of cellular junk. Thanks to some serious biochemical work, we've been able to identify its main component.
If you were to deconstruct a Lewy body, the main ingredient you'd find is a protein called alpha-synuclein. Now, in its day job, alpha-synuclein is a perfectly normal protein, probably helps with releasing neurotransmitters. But in Parkinson's, something goes terribly wrong. It misfolds, gets sticky, and starts clumping together.
And this happens in stages. You start with the normal single proteins, the monomers. Then they start misfolding and clumping into these small soluble groups called oligomers. These oligomers then link up into bigger thread-like fibrils, which eventually pack together to form that dense Lewy body. And here's a key point. A lot of researchers now think that it's actually those intermediate oligomers that are the most toxic, the real troublemakers, before they get locked away inside the Lewy body.
Now here's where it gets even more interesting. This whole process isn't static. It doesn't just happen in one spot. A ton of evidence now suggests that this pathology actually spreads through the brain, almost in a prion-like way, where one misfolded protein can corrupt its neighbors, cell by cell.
The leading theory for this is called the BRAC hypothesis. It's basically a timeline for this pathological invasion. It suggests the whole thing kicks off in the periphery, like in the nerves of your gut or your olfactory bulb, and then it slowly creeps its way up the brainstem. It's only when it hits stage 3, when it reaches the substantia nigra, that the classic motor symptoms finally start to show up. This model is a beautiful explanation for why those non-motor symptoms can appear so early.
So you might be asking, is there any hard proof for this spreading idea? You bet there is. This study here is one of the most powerful pieces of evidence. In some trials, patients with Parkinson's received transplants of healthy young neurons. Years later, after they passed away, scientists looked at their brains and found that those new, once healthy cells had become riddled with Lewy bodies. It's like the bad alpha-synuclein from the host brain had infected and corrupted the healthy new cells. It was a bombshell finding.
But of course, this misfolded protein isn't acting alone. It has accomplices.
So let's widen our investigation and look at the whole network of genetic factors and cellular systems that help create the perfect storm for this pathology to really take hold.
Studying rare inherited forms of Parkinson's has given us incredible insight. Of course, you can have mutations in the gene for alpha-synuclein itself, SNCA, which makes it more likely to clump. But other key mutations are found in genes like Parkin and LRLK2. And what do? They mess with the cell's garbage disposal systems, the machinery that's supposed to clear out misfolded proteins. If your garbage disposal's broken, trash is gonna pile up. And these genetic clues point to much bigger problems. You get dysfunction in the mitochondria, the cell's power plants, leading to an energy crisis. This stress then cripples the lysosomes, the cell's recycling centers, so they can't clear out the bad alpha-synuclein. All this chaos can then trigger chronic neuroinflammation from the brain's immune cells, which just adds fuel to the fire and accelerates the damage. It's a vicious cycle.
And this brings us to a really critical point. Where these Lewy bodies pile up really matters. In classic Parkinson's, the accumulation is heaviest in the brainstem and basal ganglia, which is why you see motor problems first. But in a related disease, dementia with Lewy bodies, or DLB, the pathology is widespread right from the start, hitting the cortex hard. And that's why in DLB, dementia shows up early. It's the same suspect, just a different crime scene with a different outcome.
Which leaves us with one final, and I think pretty profound question that the field is still wrestling with. For years, we saw the Lewy body as the villain, the thing that kills the cell. But what if that's wrong? What if the Lewy body is actually a protective tomb, a last-ditch effort by the neuron to wall off those smaller, truly toxic oligomers? Is it the murder weapon, or is it a containment shield? Answering that could completely change how we think about treating this disease.